Hi, my name is Annica Stull, and I'm an MD PhD student in the lab of Renee Solis at UC Davis. We study invasive non-typhoidal salmonella disease, and I'll be talking about host microbe interactions in terms of neutrophils and salmonella and how pathogenesis occurs and how we can inform better treatments. So what is invasive non-typhoidal salmonella disease? Generally, salmonella disease involves diarrhea. However, invasive salmonella can occur when salmonella crosses the gut and disseminates into the blood and lymphatics and then seeds at systemic sites like the liver and spleen and replicates there. And this leads to a severe febrile illness that results in 20 to 25% mortality. There are 3.4 million cases a year globally. Part of the reason why the mortality rate is so high is due to antibiotic treatment failure, and this relates to both host factors and pathogen factors. I focus on the host factor malnutrition and the pathogen factor of genome degradation. First, I'd like to share about the findings on pathogenesis. There are two sequence types I've been studying. ST3RN3 is more prone to cause invasive disease, and ST19 includes strains that are more prone to cause gut disease. And I focus on D23580 and SL1344. I look at how these strains interact with the neutrophils, and if neutrophils are able to produce potent reactive oxygen species that lead to intercellular killing. What I found is that my invasive strain induces less ROS than my gut strain in all neutrophil models, and this included primary human neutrophils from a cell line, and mouse neutrophils. In addition, I saw that my invasive strain was able to survive at higher levels and increase in numbers over time, contributing to its severity and disease. So the question is, what are the genotypic differences that explain this phenotypic difference? What I found is that there's a known single nucleotide polymorphism in PGTE, and this leads to overexpression. And I found that this contributes to decreased ROS induction by my invasive strain. I also saw that a prophage-encoded GTRC that modifies the LPS of my invasive strain also leads to decreased ROS. What are these components? They relate to complement, which is a serum factor that binds to the outside surface of the bacteria and helps neutrophils recognize it and take it up. So in conclusion, I've found that there's both direct and indirect reduction of complement binding by my invasive strain, and this contributes to decreased ROS induction and increased survival in neutrophils, partially explaining why the invasive strain is more prone to cause severe disease. Next, the question is, how do we find better treatments for emerging multi-drug resistant strains? I look at vitamin A deficiency, which is related to malnutrition and co-endemic with invasive non-typhoidal salmonella disease. I've studied a mouse model of invasive salmonella and vitamin A deficiency and demonstrated that male vitamin A deficient mice are more susceptible to infection. And this is in the blood, liver, and spleen. I then hypothesized that vitamin A supplementation during antibiotic treatment failure would help combat invasive non-typhoidal salmonella infection in vitamin A deficient mice. And indeed, I saw that co-treatment of vitamin A and subtherapeutic antibiotics decreases systemic salmonella burden and improves survival in vitamin A deficient male mice. Importantly, this shows that vitamin A treatment during antibiotic treatment failure alleviates invasive salmonella disease in susceptible mice. It also shows that it's important to look at sex differences in infectious diseases and immunity, and we hope that this mouse model and finding can inform a clinical trial in people to help decrease mortality. I'd like to thank my lab, our collaborators, and the funding that we've received in support of this project.